Okay, I'd like to tell you something about choice and variability in the immune system this afternoon. And to do that, first of all, I need to tell you a little bit how the immune system actually works. So when you get an infection with some virus or bacteria that's entered the body and set up a beachhead, the immune system responds by rushing a lot of different cells to that area to try and attack it and contain that infection. So all of those cells, they're very diverse. There's all sorts of different types of cells, very specialized for particular functions. So some cells might make some antibodies, other cells might directly kill infected cells, other cells might eat bacteria. So that degree of specialization means you've got to have a lot of choices about what type of cell that you deploy to fight that infection. And during the response, there are also some changes that go on where one cell type will change into another more effective type. And one of the ones that we've been very interested in is the T cells of the immune response. They're one of the major arms. And during a response, those initial starting T cells don't have a lot of functions. They can't really attack the pathogen themselves very much, but they can then change into cells with much more effective functions. And they can change, for example, into Th1 cells, and those are very good for attacking bacteria and viruses. They could also change into Th2 cells, and those cells are very good if you're trying to combat a parasitic worm, but they're not so good for the virus and bacteria. So the immune system needs to make the right choice about what type of response to induce. And that's particularly important because these cells also have a downside. So these Th2 cells that are so good for parasitic worms, they can also induce allergy and asthma. So you really need to get that choice right. So we need a very precise mechanism for choosing, but at the same time, we know that cells in the immune response in various of these populations I've been describing, even within those populations, they can be very diverse. So you have this enormous amount of variability within a population, and in these scatter plots at the bottom here, you can see that the expression, each dot is one cell, the expression of different proteins can vary over a level of 10-fold or more. So that's a lot of variability with the population, and yet you want a very precise response, very precise choice as the infection proceeds. So I'm going to go through a few thoughts on what happens when you combine that choice and that variability. So what I'm going to use is what's called a landscape model to illustrate some principles. And the idea of doing this is not that our cells are really rolling around a landscape, but as people, we can visualize things quite effectively on this particular kind of image. We're all used to the idea of what happens when balls roll the landscape, so it's an idiom that we can use to try and talk about cell behavior. So what I'm going to do is say that the landscape represents a whole bunch of different states of a cell. So if a T cell is occupying one part of the landscape, that means it's a particular kind of cell. And it can change. It can move across the landscape into something else. And it will obey the laws of gravity, so it will roll down. And so a basin will tend to be a state where that cell is very stable. It's going to stay there for quite a long time. Whereas a hillside is not going to be very stable. It's going to roll down. And what I want to do is combine the idea of the balls rolling down a landscape with the variability that I was speaking about. And we set up a computer simulation that allows cells to roll down one of these very complicated landscapes, but at the same time have some random movement built in, so that the motion isn't completely controlled by gravity. There's a lot of shaking around as the cell has this random movement applied over the top. So when we do that, I'll start off with a very simple model where we simply got a tilted landscape, and over on the top, there's a basin with a whole bunch of black cells in it. And when we start it rolling, then those cells will go down the hill, and they'll generally go towards these two basins at the bottom. And some go into the green basin, some go into the red basin. And you can see that we've got this random motion programmed into it, and about half the cells go into each pot. And we can run this simulation over and over again, and about half of them go each way because we've made it symmetric. But what happens when we look at, rather than the whole population, if we look at just two cells within that population, and so I've colored two of them so you can see them, and as they start to run down, then you can see that they're also shaking around like all the black ones, and their movement doesn't really give you much of a hint about where they're going to end up. If you try to guess where they're going to go, As you get later on, you can, but for quite a while, it really wasn't clear what that preference would be. So if we run it again, just with slightly different cell positions, the same sort of thing happens. Those cells are dancing around, so the yellow one is pretty easy to predict where it's going to go. The blue one, I think, is going to confuse you a lot more, and I think he finally settles down about now, coming down, and he drops into the red one as well. So the point I want to take away from this is that the behavior of populations of cells, even with this random variability, we can predict the population behavior pretty well, but it's very hard to predict the behavior of the individual cell. So if we take that a step further and think about the effect that just variability might have on a response and on the change of state of these cells from one state to another, here I have a basin in the middle that represents a relatively stable state of cells. So cells in there are rattling around, there's a fair amount of movement, they're not all just sitting at the bottom, but none of them are escaping that state. So we say that would be a very stable state. We could push the cells one way or another to try to get them to change, but instead, I'm going to do something different. I've now turned on increased variability in the cells, so now they're simply moving more, not in any particular direction, but that has the effect that a few of them start to spill over the edges, and now you can escape to states that I've shown as the pink and the blue states off to the side that might be very different. And there's a very important feature of this diagram, and that's the idea that if you want to know what's going to happen in this model, it's no good looking at the middle of the well. So the great bulk of cells in the middle aren't telling you what's going to happen. It's those very few outliers on the fringe of the population that may be the crucial difference between whether you get some of these new cells or you don't. So I think that's something that we can certainly talk about in human populations as well. In some ways, you want to pay a lot of attention to what the great bulk of people are doing and thinking. But there are certain situations where it's the outliers that really make a difference, that will make some change that the great majority of people will not get to immediately. So we can take this analogy a step further by looking at what happens if cells have multiple paths to get to the same place. And so in this animation, I have again some cells sitting in a well at the top, and we have a target well at the bottom, and they're all going to end up down there eventually, but we've given them some intermediate states that are sort of semi-stable that they can sample on their way down. And when you start that running, these cells flow down. We've colored them. If they go through one well, they go blue, another one green, and the third one red. And as they flow down, you can see that the colors being added tell you what path the cell has taken down the mountainside. And by the time you get to the bottom, it's obvious that you have cells of pretty much any color that you can imagine from those combinations. These cells are taking all sorts of different ways to get from one state to another state. So that could be very interesting if you have a situation where you want to modify some of these pathways. And we can look now at the same animation, except on the left-hand side, I'm going to emphasize the green pathway. So the cells are going to go mostly off to the right, and that's a dominant pathway. That seems to be the main way that cells get from the top to the bottom state. But on the left, on the right-hand side, I've changed one of those bowls, one of the attractors, I've changed it into a mountain. So the cells will not be going in there, they'll be deflected off of that. So when you run that, you can see that in fact happens. We get a majority of cells going in the green direction in one case. When we block the green direction, the cells simply find other ways down the mountainside, and we still end up with similar numbers of cells in the final state. It's just that they've got there in the majority of a different way. So that may be very important, for example, if we have an infection. Many pathogens interfere very strongly with the immune response, and they'll block different pieces of the immune system. So it's very good to have a lot of flexibility so that you can work around roadblocks that can be put up by the pathogens. The other way around, it may be a little more awkward if you're trying to identify a dominant pathway and produce a therapeutic drug to block that. You may be surprised that the immune response works around that as well. So I'd just like to summarize the main 3 points I tried to make today. The first one, that populations can often be, their behavior can be often predicted quite well, but it's very difficult to predict the behavior of these individual cells. that make up the populations. And so if we really want to understand how the system works in detail, we have to make more and more measurements at the single cell level, which is a lot more difficult than measuring population behaviors. The second point was that variability in and of itself can initiate changes in the population, and you can go from what appears to be a very stable situation to a movement into a different state just by increasing the variability without inducing any other change. And it's an interesting point, I think, that for some purposes we may wish to look at diagnostic methods and say, when should we be looking at bulk results, and when should we be actually looking at the variability in the population as well? And I think the point about multiple paths to the same goal may be very important, as I mentioned, for the flexibility of the immune response in different directions. And I wanted to close off, though, by just pointing out that everything I've told you so far is fantasy. So I've used this landscape model to try and make some conceptual points about what happens when we consider the interplay between choice and variability. And I think now this might give us some interesting ideas to follow up in more experimental approaches. Thank you.